good to go.
Yeah.
It's Spirea is an antibody drug conjugate company. So I will do a brief introduction to what ADCs are. Always not required, but anyway, I'll do it. So they're targeted therapeutics. So it's a combination of an antibody with a therapeutic payload linked with a linker. The goal, obviously, is the magic bullet, is to guide the therapeutic payload to the target cell, almost always the cancer cell. The ADC is internalized, the payload is released, and it does its job, which is, generally speaking, to kill the cancer cell. Nothing more complicated than that. Our proprietary technology is a linker technology. And so really the right-hand side of the slide is probably the most important thing from this slide. I mean, most people think of linkers as something which simply links the payload to the antibody. They do way more than that, and it's become more evident over the last couple of years. They can influence ADC stability. They have you can dictate the rate and where the payload is released, which is obviously critical to the ADC. Particularly relevant to us is the number of payloads that you can attach to the antibody, and also the PK. So that's also a very important part of what the linker can do. So you'll probably guess by now that we are a linker company. Oh, that's not fully on the screen. Okay, so so we're a linker company. We have a proprietary patent-protected ADC linker technology. The offering, if you like, to the field is that this linker will enable the use of novel payloads such as degraders and dual payload ADCs. So currently the field is very focused on using a very small number of payloads, very potent toxic payloads, and that has issues in terms of resistance and toxicity. Right, we're getting there. The technology is a linking technology, so it's completely independent of the target or the antibody that you're using. So it's a great platform that can be used in lots of different settings for different disease indications, etc. And obviously, we're looking to generate unique next generation therapeutics. So partnering is a key part of what we're doing. There's lots of potential payloads out there. There's lots of potential targets out there. Lots of indications. We can't cover it all, and there's lots of companies working on ADCs, so we want to try and exploit those synergies as much as we can to create more therapeutic opportunities, greater value generation, and for us, of course, ultimately, potential M&A opportunities. And needless to say, the goal is to treat more patients and improve survival, but not just improve survival, but also improve their journey towards survival, because currently, you can say that ADCs are fundamentally still chemotherapeutic agents that suffer a lot of toxicity issues. So it's a bit of a rough ride being treated with ADCs as it is with chemos. Right, so this is the technology. So it's a polymer backbone. So we have a polymer backbone which we can extend in a very controlled manner. And depending on how much we extend it, we can attach more or less payloads to the polymer backbone. So we can have any number of payloads we want. We aim for 4 at the moment. It can be more or less than 4. The linker payload combination is then conjugated to the antibody in a stochastic way. So we can increase or decrease the DAR, the drug to antibody ratio, depending on what we want. It includes, so this payload subunit includes the payload, obviously, the release mechanism, and also a hydrophobic shield. Because one of the key issues with ADCs is if you put too many payloads onto the antibody, you're coating the antibody with a hydrophobic molecule. They aggregate, the PK is a bit of a disaster. So there was, historically, there was this contradictory result. The more payloads you put onto an ADC, the worse its performance. And performance was going down because the PK was suffering. So it's a very flexible system. We can adjust the number of payloads to suit the indication. We haven't done it yet, but theoretically we can put more than one type of payload on the ADC, which is also a very hot area at the moment. People are quite keen to explore that. So as I say, a very flexible system which can address some of the ongoing issues with ADCs. So as I say, the field is kind of locked into a particular type of ADC at the moment, high potency, high toxicity payload ADCs. The drug to antibody ratio is capped at 8. As I say, mainly because of aggregation issues and poor PK. If you try and go higher than a DAR of 8 without mitigating for that hydrophobicity, and almost all ADCs at the moment are a single payload, say limited to 1 or 2 or 3 different mode of action payloads, which is a dangerous place to go because if the patient develops a resistance to that particular payload mechanism, then there's very few other places they can go in terms of being treated with an ADC. I think I've really outlined where we differentiate from the current normal scenario, if you like. So we've generated quite a bit of data. So we know, and this is very important, we know that we can generate ADCs of a particular DAR, so very focused DAR, so we don't get lots of different species from our generation of ADCs. We have great stability, so stability is very important. Stability is quite complicated actually, because you kind of want the right stability. It seems obvious that you want a lot of stability in order to make sure that your ADC reaches the target tumour cell intact. But you don't want too much stability that it's never actually released when it gets there. But if it's not released when it gets there, it actually keeps on going around in circulation, and actually we actually get more toxicity issues rather than less. And when we have the right level of stability, if you like, with our ADCs, the linker payload does not affect the performance of the antibody, which is obviously quite important. We want to make sure that the antibody is still binding to the target. We have great in vivo efficacy, and we have lots of data which we can potentially show you after the, if you want to talk to me afterwards or want to see a more full presentation. So we get complete tumor regression in mouse xenograft models, both CDX models and PDX models. So that's working very well. Pharmacokinetics is great. So the pharmacokinetics is pretty much the same as the naked antibody, which is kind of the holy grail if you can achieve that. And as I say, the lack of aggregation, so the lack of aggregation is directly related to the PK, and the performance of the PK is obviously related to the effectiveness of the molecule. And we get good rodent tolerability, so that's really linked to the stability. If the ADC is falling apart too early, then the rodent will suffer from toxicity issues. The market is expanding. It's expanding rapidly, even with the limitations of these 3 or 4 different mode of action payloads. But I think our pitch to the field basically is payload flexibility. So as I say, there are a limited number of payloads and all high potency payloads. If you can find a way to attach a payload to an antibody or any payload to an antibody which is low potency and still maintain efficacy, there's a whole universe of payloads that you could potentially address. So we're using the antibody market as the naked antibody market as a proxy for that, and I think that makes perfect sense. You can still have a functional antibody, but if you provide it with an additional warhead to increase its activity, then you're going to give that extra activity. So I think the ADC market, if you can really get to that magic bullet scenario, then it could be much bigger. So these are some pretty nice exits recently. So there's been one only about a month ago. So companies, whether it be pharma companies or maybe CDMOs, are looking for platforms that they can bring in-house, that they can address the ADC market and all that interesting novel payloads and novel targets. So yeah, some nice exits there, and many of them are based around the platform. So Lonza Genetics is a classic example. Synaffix didn't have any pipeline. They had a very nice suite of ADC platform technologies, and they were acquired by Lonza, and now Lonza offer that as part of their ADC development work. So why invest in Spirea? So I've just described the fact that we're going to enable a key advancement in ADCs, i.e. the move to novel payloads, different types of payload models, breaking away from that limitation of these super toxic payloads and that limited fact that the field is limited to addressing late stage cancers. And also address a much broader range of diseases. And it doesn't necessarily need to be limited to cancer. If you can find a way to maintain the efficacy of an ADC, even with a low potency payload, then you can be looking at inflammation, anti-infective, all sorts of different fields. And some other companies are looking at that now.
I know you've got some very strong collaboration with Big Pharma. Are you allowed to talk about those today? Is it kind of
a little bit? Yeah. So we have a couple of proof of concept evaluations ongoing at the moment with big pharmaceutical companies. So they come to, all these collaborations on payload basis. So it really fits in with what we hypothesize. So they're coming to us with novel payloads, low potency payloads that can't get the efficacy they need, so we decide that if they increase the drug to antibody ratio while maintaining PK, then they will then they can really break into a new field. So two big pharma collaboration or evaluation proof of concept collaborations. I'm hoping they will convert to license agreements, and we're in discussion with 3 other companies as well. One pharma and a couple of other biotechs. So there's a lot of interest in looking at this and seeing if we can get.
Do you like to mention the names of the companies, or
no? They're fairly.
They're big American.
So we've got a big American. We've got.
Big job. No, but I think it's great that this is Spinout from Cambridge. We invested like 5 years ago or something.
Yeah.
They managed to do some like very credible deals with large pharma. It wasn't a hot area when we first invested, I have to say, but now this is a super hot space, and I'm sure that they'll have good.
I get it for some money at a time, so we're raising 4 million. Probably ought to just very briefly tell you what we're going to do with it. So that the 4 million is to further exemplify and build that platform. So there are lots of things we could do. We have a next generation payload, which is not one of those families that I've just talked about, which could be very exciting. We want to look at dual payload ADCs and degrader ADCs. The degraders were mentioned briefly earlier. So degraders have their own issues with getting into cells, and they also have issues with creating degrader antibody conjugates. So we'd like to look into that. So we're working with a small Chinese company to try and create some degrader antibody conjugate companies. And so this will be a very exciting package for 2 reasons. One is I think it's going to attract even more interest from pharma companies, but also for ourselves. We'd like to use that as a launch pad to kickstart a pipeline using our.
And do you think during your funding rounds, that rather than raising 50, do you think that there's enough sufficient interest in the space and your technology that one of the big guys will probably just snap you up immediately?
Yeah. Synaffix is that was a CDMO acquisition, but no, you saw Tahoe taking out Aravis, Synaffix being taken out by Lonza, and Maverick's taken out by Lilly. So I think that's why we're in a bit of a hurry because it's quite a hot area at the moment. So if we can put this data package together very quickly, I think that would be very exciting for acquisition opportunities.
Any questions?
How will that Zoom?
Well, we are talking to 1 or 2 CDMOs. Whether
I just wanted to ask you a question about the payloads for your in vivo study that proved such a good efficacy. What payload did you use?
So as a proof of concept, we've been using topoisomerase. So that's a bit of a contradiction, because I know that's one of the key family that everyone else is using. We wanted to make sure that we were using a low potency version of that. And we do have our own antibodies to a particular target, and there was a competitor program using a auristatin approach. And we've shown superiority. So we know
with the topoisomerase.
we want to go one step further. So that's like a stepping stone. What we'd like to do is use a lower potency, novel mode of action, or new mode of action ADC, and jump over the top out into another approach.
Okay, thank you so much.
Yes, no problem.